Welcome back to The Explainer. I am so incredibly excited to have you here with us today. You know, as a science communicator, there is literally nothing I love more than unraveling the hidden mysteries of human biology. Today, we're diving into a really fascinating genetic puzzle about how our DNA actually works on a day-to-day basis. We tend to think of our genes as simple on and off switches, right? But the reality is this brilliantly complex system of checks, balances, and critical fail-safes. So let's jump right in and look at the blueprint of life itself. To really understand our genetic blueprint, we have to start with a fundamental truth about human biology. We are diploid organisms. That means we inherit two copies, or alleles, of every single gene, one from mom, one from dad. I want you to imagine these two copies as two identical side-by-side assembly lines in a bustling factory, both working around the clock to pump out the proteins your body needs to survive. Now, under perfect conditions, both assembly lines are just humming along. But the critical question we're exploring today is, what happens if one of those assembly lines breaks down? What actually happens when your genetic backup plan fails? Section 1, the genetic backup plan and the rule. Okay, to answer that question, we first need to establish the baseline rule of genetics. Nature is incredibly efficient, and for the vast majority of our genes, it has designed a highly resilient backup system. And this brings us to a wonderfully elegant biological concept known as haplosufficiency. Haplosufficiency, I'll just pause for a second to let that term sink in. Breaking it down, it describes a situation in diploid organisms where having just a single functional copy of a gene is perfectly sufficient to maintain normal physiological function. Going back to our factory analogy for a second, if one assembly line suddenly shuts down because of the genetic mutation, what we call a loss-of-function variant, well, the other assembly line just rolls up its sleeves and handles the cell's full production quota all by itself. The body literally doesn't miss a beat. So the crucial takeaway here is that because the vast majority of the genes in the human genome are haplosufficient, loss-of-function variants in these genes are typically associated with recessive phenotypes. In plain terms, it means an individual won't show any symptoms of a disease unless both alleles, both copies of the gene, are broken. If you have just one working copy, you are protected. We have a really classic example of this rule in action, cystic fibrosis. As you might know, this disease is inherited in an autosomal recessive manner, and it's caused by pathogenic variants in the CFTR gene. Normally, this gene produces a protein that regulates the movement of chloride ions, so salt and water, across cell membranes. When both copies of the gene are mutated, the body produces a defective protein, and that leads to a buildup of thick, sticky mucus in organs like the lungs. But because the CFTR gene is haplosufficient, a person with just one damaged copy and one totally healthy copy produces enough functional protein to maintain perfectly normal physiological processes. These folks are carriers, and they remain completely asymptomatic because that one good allele adequately compensates for the loss. Nature's backup plan works flawlessly.
Section 2. The exception. Haploinsufficiency.
But, biology is never without its twists. What happens when the factory's backup plan simply isn't strong enough to carry the load? We're now going to shift gears and look at the exception to the rule, a genetic reality where a single working copy is a massive problem. This exception is called haploinsufficiency. Haploinsufficiency. Notice the subtle difference in the word there, but trust me, it represents a completely different ballgame in human biology. This is a situation where having just a single functional copy of a gene is completely insufficient to maintain normal physiological function. The single assembly line just cannot produce enough protein to keep the factory running smoothly. Because the remaining unaffected allele can't maintain effective function on its own, a loss-of-function variant in just one allele produces a dominant phenotype. One strike and the disease is present. Now, what's really fascinating here is exactly how rare this exception is. To put it into perspective, we have roughly 20,000 protein-coding genes in the human genome, and as you can see, only about 15% of them are estimated to show haploinsufficiency. The vast majority, that 85%, follow the safe recessive backup plan we just discussed. But for that 15% slice, the stakes are incredibly high. These are often genes that act as master regulators, where the exact precise dosage of a protein is absolutely critical to survival.
Section 3. Case study. Dravet syndrome.
To truly grasp the real-world gravity of this genetic exception, we need to look at how it affects patients with devastating conditions like Dravet syndrome, which is a severe form of epilepsy that begins in infancy. Now, you might be wondering, why are some genes haploinsufficient in the first place? Well, very often, it comes down to what we can call the subunit problem.
Many haploinsufficient genes actually code for proteins that are just single subunits of much larger, complex protein structures. It's like building a car on an assembly line. If the factory doesn't produce enough steering wheels, you can't just build a functional car with a fraction of a steering wheel. The whole car cannot be finished, right? In the body, the underproduction of a specific subunit reduces the total number of fully formed functional protein complexes the cell can even use. Okay, let's walk step by step through the physiological cascade of Dravet syndrome. Step 1. The patient has a loss of function variant in just one allele of the SCN1A gene. Step 2. Because of this single broken copy, the body cannot produce enough of a highly specific protein called the NaV1.1 alpha subunit. And this subunit is a supercritical piece of a sodium channel in the brain. Step 3. Without enough fully formed NaV1.1 sodium channels, the brain simply cannot properly initiate and propagate electrical signals, which causes a severe disruption in the balance between excitatory and inhibitory signals, which leads us to step 4. The individual becomes highly prone to debilitating seizures, all because one single gene copy couldn't produce quite enough of one specific part. And this comparison right here brilliantly illustrates the core difference we've been unpacking today. On one side, we have a recessive phenotype driven by the CFTR gene in cystic fibrosis. One allele is damaged, sure, but the other compensates perfectly. On the flip side, we have a dominant phenotype driven by the SCN1A gene and Dravet syndrome. One allele is damaged, and the remaining allele simply cannot maintain function alone. It is a stark, absolute contrast between a genetic system with a built-in safety net and a genetic system balancing on a tightrope. Section 4. Rethinking genetic dominance. Understanding this biological twist fundamentally changes how we view inherited traits. I mean, we aren't just looking at a simple dominant trait like having brown eyes versus blue eyes. We're looking at a vital biological threshold that completely challenges our conventional understanding of genetics. Let's synthesize this with our critical takeaways. Haploinsufficiency proves to us that certain genes strictly depend on having both functional copies to preserve normal physiological functions. And these aren't just any genes. They're typically the ones with incredibly vital roles in our developmental and fundamental cellular processes. And because they're so critical, even a partial loss of expression, literally just a 50% drop in protein production, leads to massive, significant clinical consequences. I actually want to read this quote directly from our source material because it captures the urgency of this science perfectly. The phenomenon of haploinsufficiency challenges the conventional view of genetic dominance and emphasizes the importance of developing genetic therapies that can compensate for reduced gene expression. This isn't just academic trivia, you guys. When nature's backup plan fails, it becomes the job of modern medicine to step in. We absolutely need advanced genetic therapies, like gene upregulation or targeted gene therapy, to boost that remaining healthy allele and artificially recreate the haploinsufficiency the body desperately needs. Which leaves us with this final thought. We now know for a fact that our genetic backup plans aren't always foolproof. So as we look to the horizon of biotechnology and precision medicine, how exactly will these entirely new therapies rewrite the rules of genetic dominance? Can we eventually cure conditions like Dravet syndrome by simply telling that one healthy assembly line to work twice as hard? It's an incredible frontier. Thank you so much for joining me for this explainer. Keep questioning, keep learning, and I'll see you next time as we decode more amazing science together.